In this part, we will continue the discussion of chapter 6, and this will be part 2. So we'll continue now discussing chapter 6. In the last part, we reach discussing the plant parts that used in mutation breeding. As I mentioned in the last slide in part 1, sometimes when we do treatment of the seeds, not all the parts of the seeds are mutated. So some parts are mutated, the other parts are not. If we look to figure 6.2, we can see this very clear. The seed, the treated seed here on the left, and it produced 3 tillers. Out of the 3 tillers, only one was subjected to a mutation, and that part of the plant produced different plants. And we can see that all the plants from that tiller are similar, while the plants from the other 2 tillers are segregated according to the generation they are in. The second plant part that we use for treatment is pollens, or living plants or plant parts. If pollens are treated, then there is 2 chances to produce a dominant or recessive mutation. If we produce dominant mutation, we can see it in M1. While if we produce recessive mutation, and it will be producing heterozygous plants in M1, then we cannot see the mutation. When we use pollens, usually we have to make sure that we have enough supply of pollens, and the pollen's longevity is long enough till we do the treatment and regenerate the pollens. And usually, as I said in the beginning, if it is a recessive mutation, we cannot see it until there is duplication of the gamete and we produce homozygous plant. The third part that we are expecting is vegetative propagated plants. And in using vegetative propagated plants, usually radiation is more common than soaking, because I said that radiation can be directed easily to plant parts, and we can produce mutation in the part that we need. And the other points that we have to take in consideration, that we have to treat the plants where they are very active in cell division. Therefore, the early the possible is recommended, so that we can produce certain parts that are mutated, and this mutation will be or the cells that has mutation will be multiplied more and more to produce new organs. Till now, we are talking about induced mutations by radiation and by chemicals. Sometimes during tissue culture, when we do tissue culturing, instead of producing original plants, we produce sometimes some plants that are genetically different than the plants. This type of variation, we call it is usually genetic variation, and we call it soma-clonal variation or somatic cell culture mutations. These mutations can be produced in order to increase the variation of the plants and produce a new plants that has a new morphological characteristics, like, for example, disease resistance, improving yield, improving quality, and or sometimes get shorter plants or higher plants. Any character can be produced. Tissue culture mutations are reported in many crops. For example, which is corn, potato, and other crops. The problem that we get in this, that we produce a lot of soma-clonal variations that are not heritable. In this case, we cannot use it in our breeding programs, even though this type of mutation sometimes produce high number of mutation. In addition to inducing gene mutation or chromosomal mutation, mutation or radiation to induce a mutation or chemicals to induce mutations can be used for another purposes. And one of the purposes that has been used before, for example, is what we call gene substitution from alien chromosomes. You all know that sometimes if we couldn't find the gene that we need in the cultivated plants, we go to its ancestors or what we call the wild relatives to bring this gene. And this is, I will explain, an experiment that has been done a long time ago in introducing one chromosome from wild species called Triticum ampullatum to hexaploid bred wheat, and we end up at the end with a chromosomal part that has rust resistance genes. This figure, we cross two different types of diploid species, which is Triticum ampullatum with Triticum turgidum or emmer. The Triticum ampullatum was 2n equal 2x, which is 14, with tetraploid Triticum turgidum, emmer. So the cross usually resulted in triploid hybrid, which is 3x. In this case, in order to utilize this, we have to double the chromosome, and in this case, we get 6x hybrid plant species. This type of breeding, we call it bridge breeding, as I mentioned before. In bridge breeding, you see, in this case, we have diploid with tetraploid and then double the chromosome. After we get the hybrid, which is Grice and Emmer, we cross it with Chinese spring. This Chinese spring is very well known in the development of aneuploids, which is usually has sometimes it can survive if it has, for example, 2n plus 1 or 2n plus 2. Then after we cross it, we end up with cross-emmer wheat hybrid, okay, with the Chinese, and we did many back crosses which resulted in, and each time we do back cross of the cross with the Chinese spring, we make sure that we have always from the emmer hybrid we have rust resistance. We end up having a genotype that's similar to the Chinese spring plus the one extra chromosome. One extra chromosome. In order to get rid of all the genetic material in this extra chromosome that coming from the umbilicatum, because we need only the resistance and we don't need the other traits because these traits are not good traits sometimes. So we did radiation to this plant, which has 42 plus 1, and unfortunately, fortunately, we were lucky to get a 2n6x42 chromosomes genotype, only one genotype they get in that experiment that has the rust resistance, which is coming from the triticum impellifera. This part of the chromosome, so we get part of the chromosome which is homologous with the other chromosomes, and it was really producing the same traits of the Chinese spring with 2n equal 6x, and at the same time it was leaf rust resistance. This is a real experiment that happened, and this is how we can use radiation sometimes in transferring parts of the chromosome to our cultivated crops. This slide shows the 3 different genotypes that we utilize in this experiment. A has the wild grass, B is the emmer or Tethicum turgidum, and C is the Chinese spring. So we have A diploid, B tetraploid, and C is hexaploid. Why we do all of these things? First of all, if we have the variation that we are looking for in our cultivated crops, we will not do any mutation. But mutation has different uses in plant breeding other than producing variation. And it is support the conventional recombination breeding procedures, and it can produce or help the breeders in doing many things other than following the evolutionary process. The first advantage or role of mutation breeding in plant breeding is producing a new morphological or physiological changes, a mutation that can that can be utilized in selection. The other one is produce single gene mutation. And you remember we said that some of the chemicals that we use can produce single gene mutation, and these can be used as markers in marker breeding assistant. Sometimes we couldn't have variation in our quantitative characteristics, and we produce some mutations that increase the variability, and sometimes we improve the quantitative traits by this new mutation. D, producing somatic mutations. Somatic mutations are very important sometimes in vegetatively propagated plants because we need a new crops, we need a new variation, because sometimes we have the same genotype which is propagated by vegetative means, and if there is one problem, for example, or disease, it can kill all the plants. In this case, we can produce somatic mutations that can help us select and in the same time maybe produce good new varieties. And we have a lot of new varieties in citrus and in stone fruits that has been coming from a new species that has been coming from somatic mutation. Sometimes if linkage are, if the two genes are very linked, have short distance on the chromosome, then they cannot break by crossing over. Therefore, we use sometimes mutation to break it. And as I said when I explained the point mutation, we can induce marker genes. As you see, mutations are very important for breeding. It appears as one of the great tools that we are looking for, but in order to utilize it, the breeder should look for very clear and specific mutation that is not found already in the population. And the second point, which is very important, we have to have a way or a method to differentiate between the mutated and not the mutated plants, because if we couldn't discover it, if we couldn't identify it very easily, then we cannot utilize it in our hybridization or in our selection process. And the other point, which is this mutation should have balanced and stable genotype. If the genotype is not balanced and not stable, we cannot utilize it in producing a new cultivars. Therefore, the three conditions for success utilization of mutation breeding is that we are looking for specific mutants that are not available. We have a method to screen the plants that has mutation from the one that doesn't have, or the wild type. And the third one, this mutation has to be stable and balanced and producing good genotypes that can be utilized in cultivar development. By this, we are ending the chapter 6, and I will start broadcasting starting from tomorrow, chapter 7.